everyone, and welcome to our module on enzyme inhibitors. In the module on enzymes, I talk about how enzymes function in the human body, and what we're going to talk about in this module are enzyme inhibitors. And the reason inhibitors are important is because this is the way that many drugs work. And there are two types of enzyme inhibitors you need to understand as a medical student, and those are the competitive and the non-competitive enzyme inhibitors. So let's briefly review how enzymes work. Recall that enzymes are mostly proteins, and I've drawn a picture of the way you can think of them here. And enzymes take some biological molecule called a substrate and convert it into some product P, which is another biological molecule that's useful to the body. So essentially they are catalysts, and they catalyze a reaction like I've drawn down here, and they are converting a substrate S into a product P. So the two types of inhibitors, competitive and non-competitive, are going to inhibit the conversion of substrate into product, but they're going to do it in a different manner. So competitive inhibitors are like this green wedge eye I've drawn right here on the screen. They have a similar shape to the substrate S, and they occupy the substrate's binding site on the enzyme, and they essentially block the substrate. So they compete for the same site as S. And what this means, and we'll talk more about this later, is that if you put a lot of S into this system, if there's a ton of this S floating around, it can bump this enzyme inhibitor out of the way and overcome the effects of a competitive inhibitor. We'll talk more about that later. A non-competitive inhibitor works differently. It binds to some different site on the enzyme away from where the substrate binds, and it changes the shape of the enzyme so that there's no ability for the substrate to bind anymore. In other words, it blocks off that binding site indirectly by binding to some other area. This means that the S binding site is different, and it doesn't have the right shape to fit S anymore, so you can't overcome this by putting a lot of substrate into this system. Essentially, the enzyme has been changed into a form that doesn't bind S anymore. This is the same effect as if you didn't have enzyme around at all. So you can think of non-competitive inhibitors as enzyme destroyers. They essentially have an effect on the body and the system of taking the enzyme out of the equation and not allowing it to do its job anymore. In the video on enzymes, I discussed Michaelis-Menten kinetics and Michaelis-Menten plots like the one shown on the screen here. To briefly review, a Michaelis-Menten graph plots the substrate concentration along the x-axis and the velocity of the enzymatic reaction along the y-axis. And there are two important characteristics of an enzyme that can be determined from a Michaelis-Menten plot like this. The first one is the Vmax, or the maximum velocity of the reaction that can occur in the presence of that enzyme. The other important enzyme characteristic that you can determine from a plot like this is the Michaelis constant K sub m. To determine the Michaelis constant, you go to the point along the y-axis, that is Vmax divided by 2. Whatever substrate concentration correlates with that point is the K sub m, or the Michaelis constant for that enzyme. So now let's talk about how the Michaelis-Menten plot would change if a competitive inhibitor were added. If a competitive inhibitor were added, it would occupy some of the binding sites for substrate. However, if enough substrate was added, in other words, if you went far enough along the x-axis to the right, you could overcome the inhibitor and still achieve the same Vmax. What this means is that when you add a competitive inhibitor, the Vmax does not change, just like I've shown on the screen here. However, because of the presence of the competitive inhibitor, the K sub m is shifted to the right, as I've shown here, going from this point on the normal curve to this point on the inhibitor curve. What that means is that in the presence of a competitive inhibitor, the Vmax remains the same, but the Km becomes higher. Now let's talk about what happens when you add a non-competitive inhibitor. So I said before that adding a non-competitive inhibitor is like taking the enzyme out of commission. It's like removing enzyme from the system. And in the module on enzymes, I talked about how Vmax is determined by the amount of enzyme you have present. So when you add a non-competitive inhibitor and you eliminate the effect of some of the enzyme, it's like removing an enzyme from the system. So the Vmax is going to fall. So this change in Vmax right here is due to the addition of a non-competitive inhibitor. We move from the black line to the blue line, which has a lower Vmax. But notice that Vmax over 2 is the same in both systems. The Michaelis constant hasn't changed, and that should make sense to you. The affinity of the substrate for the enzyme remains the same. It's just that there is less functional enzyme around because of the non-competitive inhibitor. This means that when you add a non-competitive inhibitor, you get a lower Vm, but the same Michaelis constant. Now let's talk about how the different types of inhibitors affect the Lineweaver-Burke plot. And I discussed Lineweaver-Burke plots in detail in the module on enzymes, but just to briefly review, a Lineweaver-Burke plot plots 1 over S on the x-axis and 1 over V on the y-axis. And it takes the same data that produces a curved hyperbolic shape in the Michaelis-Menten graph, and it converts it into a straight line like what I've right here. And on a Lineweaver-Burke plot, the point where the straight line crosses the y-axis is equal to 1 over V max, and the point where it crosses the x-axis is equal to minus 1 over Km. So now let's talk about how the Lineweaver-Burke plot changes when you add a competitive inhibitor, and it's very easy to remember how the line changes if you remember which variables stay the same and which change. Recall that I told you that when you add a competitive inhibitor to a system that Vmax does not change. This means that the point at which the two lines cross the y-axis should be the same because that point is 1 over Vm, and Vm is the same whether you have a normal or a competitively inhibited system. I also told you that when you add a competitive inhibitor, Km goes up. Well, when Km goes up, then negative 1 over Km, this entire thing down here, is going to move closer to 0. And this means that the point at which the inhibited curve crosses the x-axis is closer to 0. And that point is a little bit harder to remember, but if you just remember that the Vmax is the same and that this point is the same, you can usually figure this other part out if you think it through. Now let's talk about what happens to the Lineweaver-Burke plot when you add a non-competitive inhibitor. So once again, it's easy to recognize the inhibited line if you remember which variables stay the same. So Km does not change when you add a non-competitive inhibitor. So the point where the two lines cross the x-axis will be the same. Minus 1 over Km will be the same whether you're talking about a normal system or a system with a non-competitive inhibitor. Vmax, on the other hand, does change. It goes down when you add a non-competitive inhibitor. And this is the tricky part to remember, but when Vmax goes down, it means 1 over Vmax goes up. So the point where the curves cross the y-axis will rise when you add a non-competitive inhibitor. That's because Vmax has gone down, and therefore 1 over Vmax has gone up. So on this slide, I've summarized the key points of competitive and non-competitive inhibitors. Competitive inhibitors have a similar shape to the substrate. They bind to the active site on the enzyme. And because they bind to the same site as the substrate, you can overcome their effects by adding more substrate. The Vmax will be unchanged by competitive inhibitors, but the Km will become higher. For non-competitive inhibitors, everything is the opposite. They have a different shape from the substrate. They bind to a different site. You can't overcome their effects by adding more substrate. The Vmax level decreases because remember, they essentially take enzymes out of the system by blocking their effects, and the Km will be unchanged by non-competitive inhibitors. And that concludes our module on enzyme inhibitors.